Welcome everybody to today's video. In today's video, I'll continue explaining protein synthesis, the process of protein synthesis. In the last video, which is the part 1, we were talking about the main ideas that you need to know in order to go deep into the transcription and translation. In today's video, I'm going to be talking about transcription. What is transcription? Transcription is kind of conveying a message but in another language. Okay, the message is initially information in the DNA. Okay, but the DNA can go out of the nucleus. What can go out? The mRNA. So we need this mRNA to pass the message from the nucleus, from the DNA in the nucleus to the cytoplasm, and specifically to the ribosomes and the tRNAs. So it's the transfer RNAs in the cytoplasm. Okay, so here the DNA. This is the first strand and this is the second one. Here the DNA is like telling the mRNA, the messenger RNA, to convey a message out of the nucleus to the cytoplasm. The mRNA here can understand the language of DNA, but it can't speak it. Like if we say that the DNA is speaking in English, so the mRNA can understand English. It wants to convey the message, but it speaks Spanish. Okay, and this language is in the form of nucleotides. Okay, and once the mRNA molecule here is made completely, it will detach. It detaches and leaves the nucleus via a nuclear pore. Let's imagine this is the nucleus. We have pores. They are like channels. The membrane of the nucleus. This is not the real drawing. It's like kind of this, but here I'm just want the idea of the mRNA going out, passing through the nuclear pores, okay, to the ribosomes in the cytoplasm. Okay. We have large subunit and small subunit. As we said in the last video, if you didn't watch it, please watch that video. It is important. Okay. And now, and before initiation, before even the transcription begins, we have gene activating chemicals. These gene activating chemicals are called transcription factors. What do? They stimulate histones at the gene transcription site to loosen. Okay, I'll tell you how. We said that this is the DNA, right? Okay, how is it represented? As chromatin threads, right? So this is a thread. If we're gonna see it under a microscope, we're gonna see these histones, 8 histones, and the DNA is wrapped around it. And we'll see another one, another group of histones with the DNA wrapping, another one, and this is a linker DNA. I explained the structure in a separate video. Please go watch it just to understand the whole picture, okay? So then these histones will loosen as a result of the impact of transcription factors, okay? This will facilitate the process of initiation to go to the promoter site and transcript, okay, and go along the gene in the DNA. The transcription factors bind to the promoter region here, okay, which is a special DNA sequence. As you see, it's a DNA, it's from the DNA, so it's a DNA sequence that contains the start point of the gene. Here, a gene starts, okay, and here it ends. And now the transcription, okay, will happen along this gene. The promoter is the first region, and we have a termination signal, and both are nucleotide sequence, okay? So here, what is the language that is moving everything, that is telling every part of this process what to do exactly? They are the nucleotides. This is the language that is the basic that everything around it moves for, okay? And you will see how these nucleotides can stop the process, and now I'm explaining how it begins. by the transcription factors that binds to the promoter. And I said it's a special DNA sequence that contains the starting point of the gene. Okay? Actually, this binding specifies which DNA strand is going to serve as the template strand, okay? And which one will serve as the coding strand. As you see here, you can notice that this strand is called the coding one, and this is the template. The template is used as a template. Simple, easy. I'll use the green here, or let's use the blue as they used it. Okay, it served as the template. But the coding one, why is this one the coding? Why the template is the one that conveys the messages or the nucleotides to the mRNA, but we call this one the coding, not this one. Because I will simplify it. Here, how are the nucleotides placed in front of each other? Let's say that C, okay, for example, needs a G here, okay? Let's say that C here needs a G in front of it, okay, right? Cytosine and guanine. I'm not talking about these now. I'm just focusing why is this one the coding, all right? If this was C, which are the names of the bases, okay? If this was C and this G, and we have the mRNA here, since this one is the template, so this G will become C on the mRNA. So same as the coding one. That's why it's the coding, because it's really similar to the mRNA that will go out of the nucleus, the coding one. But the template one is the non-coding one. So just to illustrate it in a simple way, this is the coding one, the coding strand of the gene, and this is the template strand of the gene, and this is the mRNA that is being formed. Let's say that here, this is C, this is cytosine. The one that before the mRNA is here, this one should be G, right? Guanine. And when the mRNA is formed, these bases will be translated here, okay, on the mRNA. Or let's say transcripted because it's the same information. So this G here will become a C. C, the same. That's why this is the coding one and this is the template one. And now everything I've been talking about is the preparation process, is the preparation of the transcription for the transcription. Now the transcription will begin. RNA polymerase, the enzyme that oversees the synthesis of messenger RNA, can initiate transcription now after these factors, transcription factors, binded to the promoter. And now, transcription begins with the initiation, all right? Here, as we see in the picture, RNA polymerase, this is RNA polymerase, binds to the promoter, okay? Pries apart the two DNA strands like this. Look how. In the next picture. And initiates mRNA synthesis. And here the mRNA will begin forming and then will elongate along the strand. And the elongation, which is the second step of transcription, I will use this color here. Elongation. Here, RNA polymerase moves along the template strand like this. See, this is the brown one is the mRNA polymerase, the RNA polymerase. Moves along the template strand, joining together RNA nucleotides complementary to the DNA. But what DNA? To the template DNA, complementary to the template DNA. It joins it to form the mRNA. And the nucleotides on the coding strand will be similar to the nucleotides on the mRNA, with an exception of the thymine. I'm not going to talk about it right now. All you need to know is that here is the template one, here is the coding one. The template one is the one that has the nucleotides complementary to the RNA, mRNA that is being formed, all right? The RNA polymerase elongates the mRNA transcript one nucleotide at a time. So it's like logically, yeah, one, and then the other one, and then the third one, yeah, like this. It unwinds the DNA double helix in front and rewinding it behind. Here it unwinds it, and here rewinds it like this in this picture. See, this is the RNA polymerase. This is the double helix. Here we have the unwinding just to separate the strands in order to form the mRNA from the strand we want, from the template one. See, this is the template one, and this is the coding strand of the DNA. All right, and here it will rewind it to save the shape of the DNA, which we know it's a double helix. The thing here, the new thing here is the DNA RNA hybrid region. It's like 16, 18 base pairs of DNA here. If you want to count it, from 16 to 18 base pairs of DNA are unwound, and the most recently made RNA is still hydrogen bonded to the template. Look, here we don't have hydrogen bonds between the mRNA and the template strand, but in this region, we still have the hydrogen bonds, but they will break and the elongation will continue. But this region where the hydrogen bonds are still there is called the DNA RNA hybrid region. This is the new thing, okay? And with our last stage, the termination, the mRNA is completed and released. The RNA polymerase, after completing its jump, is also released, all right? And here the synthesis ends. After what? After reaching this region, which is the termination signal, all right? And this region has a specific nucleotide sequence that signals the process to stop, all right? And I'll not talk about these nucleotides now or the bases now. If it was C, O, C, G, A, G, and these stuff, I'll not talk about it now. I'll talk about it in a separate video after completing protein synthesis. Because first of all, I need you to know how proteins are synthesized and then go deep into the nucleotide details. Okay? So the termination is the last stage, and now our transcription process ends. And then the completed mRNA, messenger RNA, that has now the codes, but they are still the same information. The DNA will leave, let's say this is the nucleus, will leave nucleus through the pore to the cytoplasm. But before translation can begin, editing and further processing are needed to clean up the raw messenger RNA transcript. So this mRNA that is released after the termination is not fully completed. It needs, as I said, further editing and processing. So initially, the mRNA, the messenger RNA that is released, we call it pre-mRNA is released. Let's say that this is the pre-mRNA, okay? We can spot two spots. One, let's say these are the exons, and the part that is called an intron. Of course, it's not one. Multiple introns and multiple exons. From their names, introns, intracellular, intra means in, so inside. So inside the nucleus. Here we are still talking about the nucleus. This is the nuclear pore. The mRNA is not out yet, okay? So introns will stay in, and exons, exocrine. That's why these are supposed to go outside. That is why we called it that way, okay? But why the green ones, which are the exons, need to go outside, but the blue one need to stay? Because the green one are the ones that code for the protein. The introns don't code for the protein at all, so they stay inside the nucleus and serve several other functions. Okay, I'll not go into this now. The pre-mRNA, after this process of, let's say, maturity, will become, what do we call the complete mRNA? We call it functional mRNA, and then it will leave. How does this process happen even? We have really important protein complexes called spliceosomes. These spliceosomes snip out the introns and then what? Splice together the remaining exon-coded sections. Here, and here. So now the functional mRNA will be only the exons like this, and will go outside. All right? And like that, after the pre-mRNA becomes the functional mRNA, the messenger mRNA, this messenger is ready to exit the nucleus through the pores, enter the cytosol, and participate in the next step of protein synthesis, which is the translation. And I'll be talking about translation in the third part, which is the last part. So I did the first part, which was the main ideas. Now this is the second part about the transcription, and then I'll do the translation later, okay? Thank you everybody for watching this video. Don't forget to hit the like button and subscribe for more. See you in the next video.